What I actually want to do first, and there's just a few of you here, I was hoping to be a few more. I first wanted to talk about, and you may need to turn on your mics or at least use the chat board, but as far as the lab reports go, okay, some are having trouble with the labeling and getting the labels to stick when you upload. Well, in all honesty, that's on your shoulders to get that figured out. I mean, it sounds cruel, but it's true. Okay. However, as I said before, there's tutoring hours. You can always email and contact me. But what I want to initiate is y'all networking with each other also. Okay. Listen, we're staying online, so you're going to have to adapt. And that's why I'm pushing y'all so hard. And like I say, as cold-hearted as it may sound when I say y'all have got to figure it out, it's just the truth. We don't know how long we're going to be online. Here at ACC, the administration isn't even talking about being in person except for limited or exceptions. So for those of you that did the labeling, I graded your labs finally. Sorry it took so long to grade the last labs from last 2 weeks ago. But keep in mind, I post them as a Word doc and a PDF for a reason. Okay. Last spring, when we transitioned into online, this class and another class had to do labeling, and I actually only posted a Word doc at that time, and they still adapted pretty well. So when I started posting a PDF along with it, I didn't expect to run into as much trouble as I'm seeing right now. For those of you that are here that have been able to overcome that problem, do you have anything to offer to others in the class as far as what you're doing or what you're using to make sure that your labels appear on your assignment when I grade it? Like Catherine, do you have something
I know I've
that you're doing?
definitely, I know I've definitely had, I guess, two issues with it. One of them I didn't realize, and when you graded it, you let me know. And the second one, I think it was, which was this past, I think, Lab 7. Um, it was when I was using a different Word program on an older computer that I don't usually use. I typically try to review it before I submit it. I look over a ridiculous amount of times just to make sure it's correct. And then once I do submit it, I always try to go back and view it because we do get 2 attempts to submit it.
Right.
Um,
Mm-hmm.
so after that first time that I messed up, I now just go back and make sure that whenever I do submit it on Blackboard, that it looks fine. Um, obviously, last
But
lab
have you,
I submitted it twice and it did funny things.
so have you been using the Word docs or the PDF docs mainly?
I've been using Word, and I haven't been using the 360 that the school gives or anything. I've just been using my own, or just the word processor that is downloaded on my computer.
Okay.
And up to this point, I haven't had an issue, and I've been through 2 of your semesters, and the last semester
Yeah.
was halfway online.
Yeah. Yeah. And I know for some students, it's complete opposite. Now, the reason why I started posting PDFs along with the Word docs is because one of my own kids let me know that Word's not as prominent among the younger generations. You know, he graduated in 2019 and he had completed his first year of college, and he waited until the end of the spring semester to let me know how few students use Word now. So I started posting a PDF. Go ahead and ask your question. You can turn on your mic.
I have a question for Catherine. When you're using Word, how are you writing on the document? Are you using a tablet or are you like writing it physically and then uploading it by taking a picture?
So I will admit to you right now, I am not very technologically advanced. I am an old person when it comes to technology. So honestly, it's really time consuming, but I end up doing text boxes and writing out whatever he has in bold in the text boxes and moving them around and drawing lines from the text box to the part of the photo. And it does take forever. I'm not going to lie. But
Okay.
it ends up with a really nice document that looks semi-organized. And yeah, that's what I do.
Yeah, because I've been having trouble both ways. I have a tablet that I bought because of this. So I try to write it on Word document and I have submitted before and just the words are just not, they don't look really nice. And I'm also using OneNote. I've been doing like copy and pasting from there, and I'm doing it for chemistry, and my teacher hasn't commented on whether she can open it properly or not. I will ask her. But most of my homework has been through there. But I don't know what other trouble I'm having though, because it opens up properly on my side, but it's not on the other side. So I don't know what trouble I'm having.
So I honestly couldn't
Chandler?
tell you about the
Go ahead,
word.
Catherine. I'm sorry.
I honestly couldn't tell you about like notebooks and iPads and stuff like that. I just use a computer. So that type of stuff is not in my wheelhouse, if you will.
Chandler, what did you want to say?
Can y'all hear me?
Yes.
Yeah.
Yeah.
Uh-huh.
Okay. So I do similarly what Catherine does with just using Microsoft Word. I do have a Mac, so I got Microsoft Word on my Mac, and I'll draw. I'll try and use bolder lines. I always have to double check the spacing between the pages because
Hmm.
sometimes whenever you submit an assignment through Blackboard, it'll move the pages and your
Yeah.
Hmm.
label get lost. So it's good to double check that. But if you're having a lot of issues, you can always print out the pages if you have a printer and you can label them. And in my math course, we have to scan all of our write out problems, homework,
Yeah, and I was going
and,
to mention that. Yeah.
and
Yeah.
send them through Blackboard. And Genius Pro is free. It's on the iPhone and it works great. There's a black and white filter that bolds everything, so it's easy to see whenever you upload it through Blackboard. And that's a really good alternative in case you're
What
having
type of
issues.
scanner Chandler, I'm sorry, what type of scanner are you using? Scanner app? Does it
It,
have, or is there a particular app?
it's called Genius.
Okay.
So
Yeah.
I do the same
So,
thing for my chemistry class. Some of the homework and even our exams, we have to write everything down and take a picture. So that way I can do it. But for the biology class, it's a bit hard to print everything out, and I'm not able to do that. So if I did, I would do it that way, but unfortunately I
okay,
can't.
and that's the reason for the PDF. Zelma mentions, you can save the Word doc. I gave it to you as a PDF because I had more students. Um, Thank you, Joey. Genius Cap is an orange logo. But I had more students. What they were doing in the spring was actually taking my Word doc and transforming it into a PDF doc because they had an easier time labeling the PDF. That's why I gave y'all PDF and Word. But listen carefully. What my son told me was an app called Cami, to where you can convert docs to PDF or you can upload PDF docs and you can add some labeling and do some editing there. There's minimum amount you can do with it for free. It gives you options to, I guess, purchase more. You might want to look at that, but that's something you have to look around at. Again, yeah, scanning is another one, but I didn't mention scanning because a lot of students don't have printers either. But the thing is this, everybody, and I went over this on the first day, okay? There was that link that I told you that ACC administration wanted students that are going to courses online to look at, especially first time. And I copy and pasted 2 questions, letting you know the responsibilities that were going to fall on y'all. Okay. One of them had to do with, hey, it's like an in-person class. The other one had to do with challenges you're going to face. So please understand, you're going to have to adapt. And trust me when I tell you, it's not easy for me either. It's not easy for the faculty either. Okay, we have faculty that have been teaching online since it first started, and they're encountering problems they've never encountered before. So Sherma, you said if you download it as a Word doc, then Blackboard manipulates the paper and ruins the labels. Yeah, if you upload the document as a PDF, it won't manipulate the document pages. That's why I posted it as both Word, PDF, but I do have some students that are very successful with using Word and uploading it, and it doesn't manipulate anything. But the PDF was what I noticed most students were transforming the document into. That's why I started giving you both. Okay, but like I said, if you don't have a way of editing a PDF doc, look at the app called Cami, Kami. Take a look at that, okay? And I just wanted to bring that up because I know, correct me if I'm wrong, if I'm pronouncing your name incorrectly, but is it Husna or Husna?
Yes.
Which one?
Oh no, you can
Or
pronounce it
neither?
as Hisna.
Okay, all right, forgive me, okay? I'm sorry
No,
if I
you're fine.
manipulate. My last name's Sanchez, and I'm used to hearing Sanchez. I finally gave in here growing up in Texas. So, but I hope this helps some. This was part of the reason, and probably should have initiated this earlier, but please listen to me carefully, okay? We're staying online,
Yeah.
and you're going to have to adapt. If you have a professor that hasn't said anything to you about the documents you're uploading, well, let me know what their secret is. So for your chem professor, let me know what their secret is, okay?
Yeah, I'll
But as I
definitely ask
said,
her.
okay, and I do know that scanning was an option, but again, the printer was the issue. You know, you
Yeah.
got to have the document to scan. Okay, well, I hope, let's see, we got one new chat. I can't see anything. Let me see. Let's see. Okay, shall we get to what was the information for today here, everybody? I hope that helps again. All right, let's go ahead and get going here because y'all are going to have a lot of labeling. That's why I wanted to address this and record it also. But let's get to, we're ahead of schedule, so I wanted to look at the Chapter 29 review before we go into a little bit of Chapter 30. For Chapter 30, I just want to get as far as the 5 traits for seed plants, if we can. If not, like I said, we're ahead. That doesn't even include the review day. So I completed Chapter 29, and if you look at the review questions, some of them are meant to be simple, but for some students, they're not simple, okay? So if you notice the first question, that was just trying to get my message across. Give 2 examples of gametes, okay, which in parentheses, that are produced by plants. And yes, it's meant to be an easy question. Okay, we don't tend to say gametes. It's a biological term. What do we tend to say in everyday life? Instead of gametes, what are the 2 terms? What are gametes in lay terms? All of a sudden, it's quiet. Thank you. Egg and sperm. Yeah, sex cells in lay terms, or egg and sperm. I would say everybody's talking a while ago. Now all of a sudden everybody's mute. Thank you. Yeah, and that's, but the thing is, I do come across students in '07 that need to hear that. Whether it's a reminder or you had an '06 professor that passed you along and you didn't learn what you were supposed to, we got to get that out of the way. Now, you can see I have in here next to gametes. So the next question, it's really saying the same thing over, but what do the abbreviations 2N and N mean? Okay, or you can say what is the difference? But first, what do we mean by N versus N? Yeah, diploid and haploid, right? That's what the 2 terms refer to, diploid and haploid. What is the difference then between a diploid cell and a haploid cell? Use a human cell as an example. Yeah, the number of chromosomes, right? So if we use a human cell as an example, a somatic diploid cell, which make up the majority of cells of your body, okay, a human cell, then that 2N would represent how many chromosomes? Total number of chromosomes. Yeah, 23 pairs, or total number would be 46, right? Either one is correct. Therefore, for an N cell or a haploid cell, if it were a human haploid cell, such as a haploid gamete, okay, then how many chromosomes would it contain? Yeah, 23 chromosomes, not pairs, but 23. In other words, so if we go by total, 2N is 46, N is 23, okay? And I tell my students in '06 is that the 2 stands for the number of sets of chromosomes. There are 2 sets. The N stands for the number of chromosomes in each set. There are 23 chromosomes in each set. One set comes from mom, one set of 23. The second set comes from dad. Second set has 23 also. So 2 times 23 is 46, okay? Now, how do these abbreviations relate to plant generations? And notice I gave you a hint, trait number 1. How would 2N and N relate to plant generations if you're looking at trait 1, which is one of 5 traits for land plants in general? Well, first you have to know what trait 1 is. What is trait 1 referring to? Yeah, alternation of generations, sporophytes to gametophytes. So how does 2N and N relate to alternation of generations, sporophytes and gametophytes?
The
If I were to give you,
gametophytes,
say it again?
the gametophytes should be haploid cells, that form to make diploid cells, which ends up being the sporophyte.
Right, exactly. So we have a gametophyte generation, which is we're talking about a haploid state, and the sporophyte generation, which we're talking about the diploid state. Okay, but we need one to have the other. Now, if you look here, plant gametophytes, question 3. When we're talking about plant gametophytes, are we talking about multicellular eukaryote plants or multicellular eukaryotic organisms? Yes or no? When I tell you this plant is in the gametophyte generation, is that a multicellular organism I'm referring to? Yes or no? Or is it a single cell? It's multicellular. So keep that in mind. Okay, during that alternation of generation, okay, when we talk about a plant being in a generation referred to as a gametophyte, okay, we're still referring to a multicellular organism. Well, think of it more generally. When you talk about the kingdom Plantae, okay, or plants, the kingdom of plants, okay, we always talk about plants being what? Just like animals, multicellular eukaryotic organisms. So just keep that in mind, all right? Therefore, what about plant sporophytes? When you talk about a plant and you're looking at it in its sporophyte form or generation, are you looking at a multicellular organism? Yes or no? True. False. True. Right. Okay. The question 5, a plant spore. Now this is going to get more complex in chapter 30. Okay, so we got to make sure we have this down here. A plant spore consists of a single haploid cell encased in a spore wall made of sporopolypen. Is that true or false? If we're looking at a spore and we look at its makeup, are the 2 key parts that form it? No. Okay. This one here is true. Okay. A plant spore consists of a haploid cell derived from a diploid cell called a sporocyte that is encased in a spore wall. This is going to be important because in chapter 30 it's going to get complex. For bryophytes and seedless vascular plants, we're talking about plants that are homosporous. The sporophyte develops one type of sporangia, produces one type of spore. That cell within the spore will initially go through several rounds of cell division. And if it's a bryophyte, it develops into a protonema. If it's a seedless vascular plant, such as a fern, it develops, say, into a fiddlehead. Is that making sense to everybody? But in the end, that haploid spore develops into a multicellular gametophyte. It's going to be different for seed vascular plants, okay? So this question, make sure you put an emphasis on this if you didn't know the answer was true. Okay, make sure you put an emphasis because for seed vascular plants, they're not homospores, they're heterospores. So we're going to talk about mega versus microspores, okay? Now, question 6, plant spores germinate in the blank by a type of cell division including mitosis. I should have been more specific in this question. Let's say question 6, make sure you make a little note. Let's say question 6 is referring just to bryophytes, okay? So add in bryophytes. Therefore, the plant spore would germinate into what initially? And I did say the answer just a while ago. Before it develops into a gametophyte, that haploid spore first develops into a wet that comes out of the spore wall. Looks like a bunch of interconnected tubes. Right, protonema. Okay, so make sure you add on bryophyte here. Now, says here, draw an example of a moss life cycle and give a brief description. This one down here, number 10, says list the 2 types of vascular plant. I'm sorry, I'm jumping ahead of myself here. Right here, back to 7. It says draw an example of a moss life cycle, then give a brief description of the life cycle beginning with the gametophyte and include the 5 traits. Okay, so question 7 here, when you're answering question 7, this is where you want to move over to this diagram here. Notice down here it says this figure here you can use to label the 5 traits. And then this one down here is where you can rewrite the 5 traits but applying them to the moss life cycle. So does this make sense to everybody for question 7? You want to then go on to slides 3 and 4, 4 and 5. Did I get that right? What am I saying? Yeah. Yeah, go on to slides 3 and 4 to help you answer question 7. Any questions on that one? Did you want to go over that one in more detail, or do you want to take a look at it first, and then if you have any questions, ask me on Thursday? Okay, everybody, it shouldn't be like I'm pulling teeth. It shouldn't be painful. All right, then I'll move on. I already mentioned homospores versus heterospores. We'll talk about this again. Number 9 says, create a flow chart representing the different classifications of land plants. What exactly do I want you to demonstrate in this flow chart? I created one for you in chapter 29. Give me a few characteristics that you would add to this flow chart. Yeah, you would have vascular and non-vascular. And you would start the very top of the flow chart off with what? What would you label at the very top from which land plants and charophytes are said to have evolved from? You would just put what 2-word term? Close. Not red, but green algae. Green algae. Okay? You would put how through speciation, there is apparently an ancestral form of green algae that evolved into charophytes and land plants. And then under land plants, you would start by writing, going left to right, avascular and vascular, right? Now, or non-vascular and vascular. Non-vascular plants, what's the more common term they go by when you're putting together this flow chart? You just don't want to write down non-vascular plants. You actually want to write down what's the more common term they go by. Begins with a B. Feel like we're doing that America Says show. Yeah, bryophytes. Okay. And then for vascular plants, do we need to divide up vascular plants into 2 groups? Yes or no? What are the 2 groups in this flow chart? What are the 2 groups? If you're writing it left to right, we would go from bryophytes to which of the 2 groups of vascular plants? No, you got to take a step back. Let me ask it this way. Do all vascular plants produce seeds? No. So you want to start with the group of vascular plants that do not produce seeds, which we covered at the end of chapter 29. That would be your seedless vascular plants. Okay. The last group would be then your what type of vascular plants? Simply your seed. Okay. Now under seed vascular plants, you want to divide them up into what they actually say 2 clades. What are the 2 clades or 2 groups you want to divide your seed vascular plants into, everybody? One starts with a G, the other starts with an A. Yeah, angiosperms, and the G stands for? The other group ends in sperms. Okay, gymnosperms, yeah. Okay, this is what we're going to talk about in chapter 30, okay? All right, so it's just kind of a rough review just to point some things out. Do make sure you go over this life cycle, even though it's multiple choice, trust me. Okay, I can come up with ways of figuring out whether or not you understand how the life cycle goes. And for the test, okay, if you can walk through the life cycle in relation to the 5 traits and then apply those 5 traits and walk through the life cycle for a bryophyte beginning with its dominant generation, and then walk through this life cycle of a seedless vascular plant, specifically a fern, beginning with its dominant generation, then with the multiple choice questions I ask you, you should be able to figure out where within the life cycle am I asking you a question so you can answer it correctly. Okay? Did that help at all? Did this help at all? Be honest. Thursday, or ask me. Okay, so if you didn't have a question today, but you have one later, please ask me. Okay. Let's go ahead and move ahead to chapter 30 here. Okay. So for chapter 30, it focuses on vascular plants, seed vascular plants. Okay. So in this chapter, you're going to get 2 more life cycles. All right. One of the life cycles is going to be applied to your gymnosperms. The other life cycle is going to be applied to your angiosperms. There are a lot of parallels between the two. There are also some slight differences, okay, as we go through this. First, for the overview or intro, transforming the world. Seeds change the course of plant evolution, enabling their bearers to become the dominant producers in most terrestrial ecosystems. Okay. Now, the first seed plants are believed to have evolved or originated approximately 360 million. It's a spore-like embryo surrounded by nutrients, and this is encased in the source of the nutrients is going to differ between angels and seeds. Talk. Okay, can y'all hear me? Yeah, this happened yesterday, everybody. If worse comes to worse, I'll just have to do this over again. Fortunately, this is why when I got a chance to get ahead, I did. Sorry about this. This happened yesterday again. All right, let's try this again. Okay, so we're focusing on seed vascular plants in chapter 30. It starts with an overview, transforming the world. It says evolution enabling bearers to become dominant producers in most terrestrial ecosystems. They believe seed plants originated or evolved roughly as a form of spore. Are the rest of am I cutting out the rest of y'all also besides Selma? Okay, on my end it says it's recording, so I'm going to try to keep talking and hope that it records. I'm going to hope that it records, and if it doesn't, like I said, I'll do this over again. Did y'all hear that part? If I need to, I'll do it over again on Thursday. Okay. All right, so let's see how it works out here. Let's try to tough it out. Okay, so in the end, for seed vascular plants, what you're going to have to do is separate, but also compare and contrast spores, the 2 different types of spores produced by seed vascular plants, versus comparing the slight differences, okay, in parts of the seeds between gymnosperms and angiosperms. But in general, whether it's a gymnosperm or angiosperm, okay, a seed contains a sporophyte embryo surrounded by and supported by nutrients, and this is all encased in a protective seed coat. Now, the source of the nutrients is what's going to differ between the gymnosperm and the angiosperm, and I'll try to point that out very carefully. Now, again, okay, you have to compare the parts that make up a seed to the parts that make up a spore, and then you're also going to have to compare and keep separated the methods of dispersal. Okay, because for seed vascular plants, it's wind and animals, specifically animals that you know as insects, that are involved, okay, in spore dispersal. Okay, but you're going to come to know these spores that are dispersed are actually referred to as pollen grains. So we're going to talk about pollination. But then we're going to talk about seed dispersal and talk about methods of seed dispersal. Okay, and there's going to be 3 different methods, which I'll go over in detail as we go. So in other words, with seed vascular plants, there are a lot more working parts. And if you're willing to go through the growing pains of studying it, you'll see where there are commonalities and differences, and it'll start to work itself out and clear up, hopefully. So if you look here, seed and pollen grains, okay? We didn't talk about pollen when we went over bryophytes and when we went over seedless vascular plants. But now this is where we introduce pollen grains. In lay terms, we don't say pollen grains, we just simply say pollen. All right. So seeds and pollen grains are key adaptations for life on land. All right. Now, down here in the bottom are the 5 traits I gave you towards the beginning of chapter 9 for all land plants. Alternation of generations, referring to the life cycle where a plant goes through a gametophyte and sporophyte stage. Multicellular dependent embryos, walled spores produced by sporangia, multicellular gametangia, and apical meristems. So these are traits common to land plants, but I also said that these traits would change. You could definitely apply them as is, the way they're given, to bryophytes. You can apply them to seedless vascular plants with a little bit of a modification in terms of the what should we call it when we talk about the sporophyte versus gametophyte generations, which one's dominant and which one's not. However, when we get to seed vascular plants, if you notice here, now you're given a new set of traits, but these traits, okay, are still tied in to these 5. Okay. Now there are definite modifications, and this is what I'm going to try to point out as we go, and then we'll go through each trait specifically. Okay. Now, these 5 traits, okay, attributed to seed plants specifically. Well, the first trait is reduced gametophytes. Okay. When they say reduced gametophytes, how does that relate back to the traits I gave you at the beginning of chapter 9? They're referring to the alternation of generations. Okay. The second trait for seed vascular plants is heterospory. Okay. This is tied in to this general trait that was given at the beginning of chapter 29. Okay. Initially, when I gave you this trait, we were actually mainly referring to plants that are homospores, one type of sporangia that produces one type of spore. But with seed vascular plants, they've evolved to where they're heterospores or heterospory. They have 2 different types of sporangia that produce 2 different types of spores, and these spores will develop either into a female or a male gametophyte. So now we have 2 separate sexes of gametophytes rather than one that the textbook refers to as, I think it was bisexual. I kept saying hermaphrodite because they use that term too, but bisexual or what I call asexual. So this is going to be another change as we go through. Do we know what determines the gender? As far as the spores go, physically you can look at them size, but as far as genetically goes at the level of the gene, I haven't looked at that. But they do develop female traits. So we'll talk about spores called megaspores, and those spores specifically develop into a female gametophyte that produces an egg. And then we'll talk about microspores. But the in-between, that I don't know, Kathy, as far as like in relation to human male and female or animals XX and XY. Okay? Sorry, it's not a clear answer, but. Next will be what we'll talk about for seed plants, okay, as far as traits go. It's going to have to do with ovules and the production of eggs, okay? When we go over ovules and the production of eggs, that's going to involve some of this, and that's going to involve some of this, okay? What I'll do when I get to this trait, ovules and the production of eggs, is I'll first go over an ovule in regards to calling it an unfertilized ovule, okay? And when I go over that, we'll talk about a type of sporangium called the megasporangium, a type of spore called the megaspore. Then I'll talk about an ovule that I describe as a fertilized ovule, or the textbook describes as a fertilized ovule. When we go over that fertilized ovule, we'll talk about how that megaspore is now a female gametophyte that's produced eggs. Okay, so we'll go over this. So how does it relate to the initial traits I gave you? Well, this is where we'll talk a gametophyte that's female, and we'll also talk about gametangia, although the textbook doesn't highlight them or emphasize the archegonia because the gametophyte is so much smaller for seed vascular plants. Next is pollen and the production of sperm. Now, pollen and the production of sperm, well, it's going to refer to the same two initial traits that I gave you as it did for trait 3 for seed plants. When we go over pollen and the production of sperms, we're also going to be talking about the gametophyte, but now it's going to be the male gametophyte. Okay? We'll talk about how the male gametophyte develops from what is called a microspore, produced by microsporangia. And we'll talk about how that male gametophyte produces sperm, and how that male gametophyte is part of what we call a pollen grain or pollen. The last trait for seed plants is the production of seeds. We talk about the production of seeds. Well, it feeds into this trait because I already told you part of what makes up a seed is an embryo. Well, if you recall when we went over embryos, okay, develop into what? Mature sporophytes, diploids. Except now this embryo is not going to be dependent at all on the gametophyte as far as developing within the gametophyte. Okay. Now for gymnosperms, in a manner of speaking, you could argue and say, well, it does a little bit, but it's not. So just trying to show you how these traits are modified for seed vascular plants. Okay. Now, these 5 traits: reduced gametophytes, heterosporia, ovules, and production of eggs, pollen, production of sperm, and production of seeds for seed plants. These are all adaptations that enable seed plants to survive and reproduce, not just as terrestrial life forms, but during droughts. So what is happening is plants have been evolving to where they're becoming more and more resilient, more and more capable of surviving in harsher and harsher environments. Okay? Now, because of that, also seed plants don't require water for what? Fertilization. What do I mean by that? Well, whereas for bryophytes and seedless vascular plants, the sperm that are produced by the male gametophytes and Theridia have flagella. Therefore, those two groups of plants tend to grow in moist habitats. This is not the case anymore. Now we're going to talk about how the male gametophyte, that generation, is encased in a pollen wall. Two together form a pollen grain, or what we simply call pollen. Okay, and that male gametophyte, okay, the way it's dispersed now, or the way its sperm is dispersed now, is much more directly. We'll talk about how the male gametophyte inside of a pollen grain, through the aid of wind or through insects, which are animals, okay, will be dispersed. And when pollination occurs, now the male gametophyte is going to much more directly deliver its sperm to the egg in the female gametophyte. There's no more releasing the sperm into a film of water and the sperm has to do all the work. Now the male gametophyte's going to do it. It's going to carry it over there. So this is going to be another big change. So for the first trait for seed vascular plants, reduced gametophytes, if you recall, a bryophyte's life cycle is dominated by the gametophyte stage. Okay. So when we say alternation of generation, we're talking about the life cycle of a plant. The dominant generation is the gametophyte. But for seedless vascular plants, we talked about how it's a sporophyte, the sporophyte generation, that is dominant generation during that life cycle or alternation of generations. Okay. Well, for seed vascular plants, the sporophyte generation is going to continue to be dominant. This is what I'm going to get into. Okay, but there's a complete 180 for the gametophyte. When you compare the bryophyte gametophyte to a seed vascular plant gametophyte. So if you look here, as plants continue to evolve, reduction of the gametophytes continued, and some populations of vascular plants began producing seeds. The gametophytes of seed vascular plants develop within walls of spores, within the sporangia, that are retained within the parent sporophyte. So now what is happening, okay, is what they're saying here is that the gametophyte now is dependent on the sporophyte, which is the complete opposite of what we saw for bryophytes. And I'll show you a little figure to show you the transition, okay? So this is going to be something that's going to be very different in the life cycle. When I point out the male and the female gametophyte, they're always going to be located within tissue that is part of the sporophyte. Now, the gametophyte, though, being retained within the parent sporophyte, is afforded protection due to this arrangement, okay? And also, it's afforded or given nutrients by this parent sporophyte. This is a big one here, though. If you recall for bryophytes, not only is gametophyte generation the dominant generation, but I said before that during a bryophyte's life cycle, at a minimum, you should be able to see it physically as a gametophyte versus seedless vascular plants. Its life cycle or alternation of generation. At a minimum, you normally see it as a sporophyte. Okay, the opposite generations are brief. And if you recall, for seedless vascular plants, the gametophyte was described as heart-shaped. Some people say you can see it with the naked eye. The plants we order for lab, nobody's found that I know of. Okay, but the thing is, for seed vascular plants, now the gametophyte, it's not only reduced in terms of the plant's life cycle. In other words, it now exists for an even shorter period of the life cycle, but physically is extremely smaller than even a seedless vascular plant's gametophyte. It is described as microscopic. And I'll be honest with you, in lab, the prepared slides we look at, so far we haven't been able to order prepared slides that have allowed us to see the gametophyte technically. I would say submicroscopic. Okay, so they're reduced in terms of physical size, but also the time they're present during the plant life cycle or alternation of generation. So reduced gametophytes, here's this figure. I showed you this figure in chapter 29 for bryophytes and for seedless vascular plants. Now we're adding on our seed vascular plants. But notice something for the gametophyte, okay? The gametophyte was dominant, okay? Over here, the gametophyte was reduced, but it was independent for seed. In other words, it wasn't dependent on the sporophyte, even though it was smaller in size, okay, versus here where it's larger, it's smaller in size, it still wasn't dependent on the sporophyte. However, if you look here, it says it's not only reduced in terms of overall size and time it exists during the life cycle, okay, but now it's dependent on sporophyte. So we have a complete 180 from here to here, because if you look at the next level, sporophyte, for bryophytes, if you go straight from bryophytes to seedless vascular plants, for bryophytes, it's the sporophyte that is reduced and dependent on the gametophyte, but we have a complete 180 for seed vascular plants, a complete 180, okay? Then from there, you can see, just like with seedless vascular plants, the dominant generation for seed vascular plants is sporophyte generation, meaning that when you're looking at a seed vascular plant throughout its life cycle, at a minimum, you should see it exist throughout the majority, if not the entirety of its life, as a sporophyte plant. So down here, they're showing you, we divide them into two groups, gymnosperms and angiosperms. And I'm going to refer to these in a second. But do take a look at this table and notice some of the terms they're using. Just for gymnosperms. No, they're not saying gametophytes anymore. Now they're saying female versus male gametophytes in completely separate structures for gymnosperms. For angiosperms, it's going to be much the same. Okay, although we may not say completely separate structures, but again, you see they're trying to show you. And in both cases, they're pointing out that they're referring to the flower of angiosperm. So this is something we'll talk about as we continue. So the second trait, so please notice on the PowerPoints, I try to do a good job of numbering the traits so you know what we are during the 5, okay? Head, the rule among seed plants. The ancestors of seed plants were likely homospores. Okay? They produce one kind of spore that usually develops into a bisexual or asexual gametophyte, okay? Now, if you recall from mosses, the life cycle, I did show you a figure, okay, where coming off of the rhizoid, you could have an apical meristem develop into a female gametophyte or an apical meristem develop into a male gametophyte. Okay, but they're all part of that one moss plant. Now, I didn't look into the genetics of it all and what results in that differentiation of that one moss plant, but it's one plant. Okay, now we're going to start getting structures that are starting to separate more for seed plants. Seed plants are heterosporous because they kinds of spores produced are tied into the fact that seed vascular plants have 2 different types of sporangia. So when you're looking at a seed vascular plant, okay, that is a sporophyte, all right, that sporophyte will develop 2 types of sporangia, usually in separate structures. Okay, and we'll go over this. If it's a gymnosperm, we're going to focus on conifers called pines. And we'll talk about how pine trees produce 2 types of cones, ovulate cones and pollen cones. And we'll talk about how ovulate cones are where you will find megasporangia, pollen cones are where you'll find microsporangia. But when we go over angiosperms, we'll focus on the flower. We'll focus on the carpel and the stamen. Okay, and we'll talk about in a certain region of the carpel, this is where you'll find megasporangia. In a certain region of the stamen of the flower, this is where you'll find microsporangia. Now, what's the big deal about these 2 different types of sporangia? Is megasporangia produce spores called megaspores. The megaspores will develop into female gametophytes, whereas microsporangia produce microspores. And these microspores will develop into male gametophytes. Okay, so even though they both are produced by the one same plant, they're still produced separate of one another. They are not physically connected at all, these 2 gametophytes. Is that clear to everybody? There's no physical connection outside of the sporophyte that they're dependent on. Okay, and I'll show you this. So one plant can sexually or asexually reproduce, but the actual gametophytes themselves, there's no tissue connection between the 2, even though they're being produced by the same sporophyte. Okay, that's in contrast to your bryophytes and your seedless. So here, as I said before, when we go over gymnosperms, we're going to focus on conifers. There's 1 of 4 groups of gymnosperms. And for conifers, we're going to focus on pine trees. And how pine trees, we know them as pine cones, but the cones you see during the holiday season, they're actually the ovulate cones, the female cones of a pine tree where you'll find a female gametophyte developing. If you ever go see a pine tree and you catch it at the right time of year and you look very closely, you'll see these really tiny, tiny cones that are called pollen cones, and they don't stand out, but they're very tiny, small cones. And these pollen cones contain pollen sacs, but you're going to know them as microsporangia. And we'll talk about how these microsporangia produce many pollen grains that contain microspores that develop into male gametophytes. And then we'll talk about angiosperms when we get there, about what's similar and what's different between these, where you find these female and male gametophytes. Okay, the third trait, ovules in the production of eggs. First of all, what I want to go over is what is an ovule, but I want to talk about it in stages. Okay. When we talk about an ovule isn't set in terms of its structure. Okay. If fertilization occurs, the ovule itself will go through a transformation process. Okay. And if fertilization occurs, not only will the ovule go through a transformation process, the ovule will become the seed. Okay. But initially, when an ovule is produced, let's say it's a gymnosperm. Here they're showing you a female cone known as an ovulate cone, and they've taken a longitudinal section, top to bottom. Okay. All the brown stuff here, okay, is sporophyte tissue. But instead of leaflets and leaves, we have what we call megasporophylls here. Okay. These little yellow dots, that you see here, here, and here, they've taken one of these little yellow dots here and they've magnified it. This is what they're showing you. That yellow dot is a megasporangium. Okay. Well, that's only part of what is an unfertilized ovule. So an ovule, when it's first formed, what they refer to as an unfertilized ovule, I put it up here what it consists of. The initial ovule, say in an ovulate cone of a pine tree, it consists of one or more protective layers of integument wrapped around a megasporangium that has produced a megaspore. Okay, and this ovule will have a small opening called a micropyle. So this right here, is our unfertilized ovule, the initial ovule that is formed, in this case, within a gymnosperm pine cone, called an ovulate cone. The integument is actually the sporophyte tissue that sometimes they refer to as a megasporophyll tissue, but here, it's the integument is here, and I tell you down here, it's a layer of sporophyte tissue that is wrapped around one of two types of sporangium, known as our megasporangium, which is actually part of the sporophyte tissue. This megasporangium, as I said before, is going to produce a megaspore through a type of cell division that involves meiosis. Therefore, unlike the sporangium and the integument that's part of the sporophyte, which is all diploid tissue, okay, the megaspore is haploid. It's a haploid cell. And then notice something, because we're going to use this to help us know where about we are located within the ovule. Notice the spore wall is this thin greenish blue line. Okay, that's the haploid cell. So this whole structure here is our megaspore, our haploid spore cell encased in a spore wall made up of sporopollen produced by the megasporangium. So all this together is an ovule. Okay, but third trait is ovules and production of eggs. All I've talked about is ovules. I'll let you look at this on your own time here. Well, this is where we get to the production of the egg part. Okay. As I said, an ovule will transform. So what can happen to that initial ovule that's produced in, say, the ovulate cone of a pine tree, that female cone of a pine tree, is what can happen is, if fertilization occurs, this unfertilized ovule that is now a fertilized ovule is going to go through some changes. Well, one of the changes that occurs actually occurs before fertilization occurs. If you recall, I said key in on the spore wall. Going over here to the unfertilized ovule, I said this here, right here, is our megaspore. Okay, but if you follow the spore wall over here to this side to where it says fertilized, before fertilization occurs, if you recall, as I said on the previous slide, the megaspore will develop into a female gametophyte. Okay, so that megasporangium that's part of the ovule wrapped in the integument that produces a megaspore, that megaspore before fertilization occurs will develop into a multicellular but microscopic female gametophyte. And it's that female gametophyte that then is going to produce the egg. Okay? What you don't see is either on that same, say, pine tree or on a different pine tree, there's a pollen cone where pollen grains are produced. The pollen grain consists of, okay, a microspore that develops into a male gametophyte. Now, I'll go over this part. It's going to be broken up right now, but what they're showing you here is when they say fertilized ovule, what has to happen, okay, is that the male gametophyte physically has to make its way over to the ovule, okay, on the ovulate cone. And when that male gametophyte physically makes its way over to the ovule, okay, it's got to reach the female gametophyte by going through the micropyle, which they show you here to here. What happens is when the male gametophyte makes its way through the micropyle, notice that opening called the micropyle closes. Now, the male gametophyte will then develop a tube called a pollen tube that extends out and into the female gametophyte, and that tube is used to discharge sperm. So there's no film of water that's needed. Now the male gametophyte is literally getting up to and close to the female gametophyte and fertilizing the egg produced by it by releasing sperm. Okay, but the fertilized ovule part itself, I try to highlight that stuff mostly in red. The female gametophyte, the egg that's being fertilized, the spore wall, and notice the female gametophyte, when that megaspore develops or transforms into a female gametophyte, okay, it's still within the megasporangium that's within the integument. So up here it says megaspore develops in the female gametophyte within the megasporangium, and then the female gametophyte produces an egg while still enclosed within the spore wall. I forgot to read that. So here's that spore wall here. Here's a spore wall here, except here it's a megaspore. Over here it's developed into a female gametophyte. Okay? So basically what they're showing you here, what I highlighted in red is related to, everything I've highlighted in red here is related to the fertilized ovule. But obviously down here, this information is what's tied into the fertilized ovule. Okay, so this information that's here, we go over it in detail when we go over the fourth trait. Okay, pollen and the production of sperm. So just to read through some of this first. Microsporangia produce pollen grains, okay, and the pollen grain consists of a microspore enclosed in a pollen wall, and that's what I'm showing you over here, okay, partly over here. Little Mickey Mouse ears of the pollen grain, okay. Listen carefully, you can write this down if this helps any. Microsporangia are also known in lay terms as pollen sacs, okay. So microsporangia in lay terms are known as pollen sacs. So you have this microsporangia, and inside of it, many sporocytes are developing, okay, into microspores. But they're developing into microspores, each within their own pollen wall. Now, the microspore itself, within the pollen wall, will develop into a male gametophyte. So what I'm showing you here, they describe as a mature pollen grain. Okay, if it were an immature pollen grain, then we would be showing you the microspore within the pollen wall. But when they say mature pollen grain, what they're saying now is the microspore developed into a male gametophyte, but it's still encased in that pollen wall. So over here, this is actually from a slide you would have looked at in lab had we met in person. The little Mickey Mouse ears that you see here, I always tell students Mickey Mouse ears and they look at me like I'm crazy. That's actually the pollen wall that's cracked open. The pink little thing here, that's actually the male gametophyte if it's a mature pollen grain. Okay. Now what will happen with, or actually down here, if you look, the pollen grains, pollen wall, okay, just like a spore wall, is made up of spore, pollen, and for protection. Now, the production of the pollen grain eliminates the need for a film of water because the pollen grain itself can be dispersed over great distances by wind or animals. Okay, and I put this down because a lot of people don't know. Insects are classified as animals, and we will talk about them when we go into animals. Okay, so again, we don't tend to say pollen grain, we tend to say pollen. Okay, now pollination. Pollination is fertilization. Okay, it can be asexual or sexual. But pollination is a transfer of pollen to a part of a plant containing the ovule. So the pollen grain, if it's a mature one, will have the male gametophyte that produces sperm encased in a pollen wall. And now it's reliant on wind or animal to be dispersed because the male gametophyte is more directly going to deliver the sperm. Now, if a pollen grain germinates, okay, it gives rise to a pollen tube that discharges sperm into the female gametophyte within the ovule. Well, what do I mean by germinate? This right here. this right here, okay, this is the body of the gametophyte, and then this long thing here is the pollen tube. And if you notice over here, there's a pollen tube, but it looks very transparent. But if you look at this pollen tube, you see a lot of dark stuff. The dark stuff in there, that's the sperm, okay? So again, I've highlighted this here, okay? If the pollen grain makes its way, notice they show you over here. If the pollen grain makes its way into the micropyle, okay, then what will happen is the male gametophyte, because it's a mature pollen grain by this point, the male gametophyte, which breaks open the pollen wall, will develop or germinate a pollen tube that extends out to the female gametophyte. And as they're showing you here, this pollen tube is used to discharge sperm. So there's no more need for water. There's no flagellated sperms. The male gametophyte is directly fertilizing the female gametophyte's egg. Of the seeds. If fertilization is successful, that fertilized ovule will transform into a seed. Okay? So it says here, if the egg is fertilized by the sperm successfully, we get transformation of that fertilized egg into a zygote, and then that zygote goes through several rounds of cell division involving mitosis, forming a sporophyte embryo. Okay? As that embryo is developing, if it's a gymnosperm, okay, the nutrients that ends up surrounding the embryo is derived from the female gametophyte tissue. If the embryo, okay, is associated with an angiosperm embryo, then the nutrients comes from a different source, which we'll talk about, I think it's endosperm. But if you look here, it says the sporophyte embryo surrounded by nutrients will be enclosed in a protective seed coat. The seed coat is derived from the integument. Now, seeds provide some evolutionary changes over dependent embryos. First of all, they remain dormant for days to years until conditions are favorable for the embryo to undergo germination. Germination just simply means develop into a multicellular full-grown organism. That's the simplest way I can put it, okay? They have a supply of stored nutrients or food for the embryo. Now, again, like I said, the nutrients, we'll go over it in more detail, gymnosperms versus angiosperms, but I'll show you a source of nutrients in relation to gymnosperms in a sec. And they may be transported long distances. So there's pollen grain dispersal, and then there's seed dispersal. Seed dispersal is not only by wind or animals, but also by water. So again, if fertilization is successful, okay, what happens is this integument, which is part of the ovule, it's sporophyte tissue, but it's part of the ovule. This integument will develop into or transform into the seed coat. What was inside, here and here, that megasporangium, that tissue deteriorates, okay, basically just degenerates and disappears, okay, based on the figure they're showing you here. But recall what I said, focus on the spore wall to have an idea of what you're looking at. Over here, the spore wall was surrounding a megaspore that transformed or developed into a female gametophyte. But now notice the spore wall. The spore wall has expanded to where it's up against now what was formerly the integument, but is now the seed coat. Notice this is the spore wall. Notice how much further out it's expanded. Inside of it is that egg that was fertilized by the sperm. Thus, we have our developing embryo. And then notice the color of the tissue they give you for the female gametophyte. They're trying to show you that that female gametophyte is the source of nutrients or food for the developing embryo. So in the end, the embryo ends up feeding on its former mama. Does that sound gross or something? Kind of like spiders. So if you look here, here's an actual seed, longitudinal section. We have our seed coat, which is very thin. This is the embryo, this cucumber looking thing. And this is the nutrients that was formerly the female gametophyte. Okay. Any questions? I give y'all a headache? Are y'all with me or are y'all sleeping? Are there any questions? Are there any questions? Okay. Yeah, I figured. I'm going to stop the recording, but are there any questions?